After the structure of a transcription unit, the promoter and terminator flank the structural gene in a transcription unit. The promoter is said to be located towards 5'end, that is upstream of the structural gene, and we know the reference is made with respect to the polarity of coding strand, that is 5' to 3' polarity, not with the template strand. Okay, now it is a DNA sequence that provides binding site for RNA polymerase. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strands. It is the presence of a promoter in a transcription unit that also defines the template and coding strands. Okay, so at the 5' end upstream of the structural gene, it's a promoter region. It is a DNA sequence that also defines the template and coding strands. By switching its position with terminator, the definition of coding and template strands could be reversed. Although the terminator is located towards 3' end, that is downstream of the structural of the coding strand, and it usually defines the end of the process of transcription. Now there are additional regulatory sequences that may be present further upstream or downstream to the promoter. Upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. Now, transcription unit and the gene. A gene is defined as the functional unit of inheritance. We all know that. There is term a cistron. The DNA sequence coding The DNA sequence coding for tRNA or RNA molecules also define a gene. However, by defining a cistron as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, and polycistronic P for prokaryotes, mostly in bacteria or prokaryotes. Now, in eukaryotes, we see exons and introns. The monocistronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or express sequences are defined as exons. What are these exons? The exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by interons. Interons or intervening sequences